Okay, so good afternoon, everyone, to people, all of the people who have joined this meeting. So this is our COP's second annual small talk organized this year, and like probably our 7th talk for this year. Today we have Dr. Lynn Yu from the Wellman Center for Photomedicine at Massachusetts General Hospital and Harvard Medical School in Boston, USA. She obtained her master's and PhD in electrical and computer engineering at U of C, and her research focused on developing spectroscopic systems to measure blood-related changes in a small target volume in the brain. These systems can potentially enable neuroscientists to study how the brain works and explore treatments for brain diseases in animal models. Currently at the Wellman Center for Photomedicine, her work focuses on the development and translation of a high-resolution cross-sectional imaging technology known as micro-optical coherence tomography. She has been an active member and passionate volunteer in both the SPIE and Optica chapters, both locally and internationally. And while she was here, she was the president of the U of C SPIE, and back then it was called OSA. Now it's called Optica student chapters, and she has served as an education and outreach committee member, career lab editorial board member, and student leadership workshop facilitator for SPIE. She's currently an OSA ambassador, not OSA. I guess they still call it OSA. I have no idea. Optica ambassador, and is also willing to share her experiences regarding this. This talk will be recorded, but the Q&A session after it won't be. So during the Q&A session, you are free to unmute yourself and ask questions, or just type it in the chat and I would be reading it to the best of my ability. And yeah, Lynn, here you go.
Okay, thank you so much for having me, and thank you so much for the very nice introduction. So for this session, I'm actually planning for 2 different presentations. The first half is going to be on my research, and I'm going to talk about my graduate research at the U of C. And also for the second half, it is part of my OSA or Optica ambassador role, which is to like facilitate discussions about career development and also like considerations about how to stay involved with professional societies and so on. So for the second half, I would like to open the discussion to be interactive. I think like I'm going to use the first 30 minutes for this research project, and then we will open Q&A for the technical part. And then the second half may be more interested to people who are still, who are still students or in the early career. So if that's good with everyone, I'll just like start the research talk part.
Great, that's fine. Yeah, I'll stop recording in between, pause the recording during the Q&A session in between. Yeah.
No problem. Let me just share my screen. Can you see it here?
Not yet.
I think it's freezed.
It's frozen?
It's frozen, yeah.
It just says Lynn Yu has started screen sharing. Nothing else.
Come on. It's frozen. Let me try.
Oh, I guess it's a Zoom problem. Like.
Yeah, I think it's a Zoom problem. Just let me try. Interesting. I may need to restart.
Yeah, probably. We'll wait.
Okay, I'll be back.
No. Yep, now it works. Yeah.
Okay, cool. Thank you so much, and sorry about that.
I mean, it's Zoom's fault.
Oh my goodness, yeah, it's better than like technical difficulties.
Yeah.
Okay, thanks so much for waiting for me, and thanks. So I'm going to present my graduate projects on single fiber spectroscopy for monitoring cerebral oxygen saturation in freely moving animals. It's not moving. Okay. So let's look at the background. The brain is the most important and complex organ in the human body. So it is really important to study how the brain works because it has really great significance in both the basic science research and also in clinical research for neurological disorders. Functional brain imaging is for visualizing or mapping the physiological parameters for studying how the brain works. And there are a variety of optical contrasts that can be used for functional brain imaging. Extrinsic optical contrast or the foreign contrast agents are usually introduced into the tissue for visualization of electrophysiology. The most common extrinsic optical contrasts that has been used are the voltage sensitive dye and calcium indicators. Intrinsic optical contrast, on the other hand, are the signals inherent in tissue, and those are usually related to the metabolism of brain functions. The most significant signals are scattering and absorption. So it's worth noting that the changes in the intrinsic optical signals also affect the measurement of extrinsic contrast. For example, can you still hear me?
Yeah, we can still hear you.
Oh,
yeah.
Cool, because like something blinked here. Sorry, I'll just go ahead and let me know if it doesn't work. Yeah. So for example, if we have a volume of tissue and suddenly there are more blood into that, it's going to absorb the extrinsic contrast. For example, maybe it could be like a fluorescence indicator or something. So that's why that it is really important to measure the intrinsic optical signals with high accuracy for high fidelity optical measurements in any type of tissue. So let's look at the intrinsic optical signals in the brain. The most dominant chromophores are that absorbs light in the brain is hemoglobin. And this figure shows the absorption coefficient of hemoglobin. HBO here is oxyhemoglobin, which means when the hemoglobin is bound with oxygen. And HB here is deoxyhemoglobin when it doesn't carry oxygen. From here, we can see that the shape of the absorption peaks are very different. That's why, like, by looking at the absorption features, we can figure out what's the percentage of hemoglobin that carries oxygen, and that's the parameter of oxygen saturation. Another parameter is blood volume fraction, which describes how much blood exists in a volume of tissue. These are important biomarkers in the brain because when there is brain activity, it consumes energy, and that energy is delivered by the oxygen and glucose in the blood. Therefore, by measuring these two parameters, it's a kind of indirect measurement of neural activities, and it's also important to study what's the correlation between the brain activities and the vasculature responses, which called neurovascular coupling. Scattering signals, which is the other intrinsic optical signal, are commonly assumed to be a static signal. So when we are looking at functional brain activities, usually we assume it's static. So for the intrinsic signals technologies, there are two major types. The first one is called spectroscopic system. Usually it has really high spectral resolution, but with no spatial resolution. A typical system looks like this. We have two optical fibers, one connected to the source to deliver the source light into the sample, and another fiber which is connected to the detector. And there is some source detector separation, row 1 here in between. The light that can be collected on the detector side usually goes through a banana shape. So it's not saying like light only illuminates this part, light illuminates all the surrounding tissue, but the light that can be detected by the detector goes through a banana shape. And when we increase the source detector separation, light travels deeper into tissue, which means the light that we sample here is a measurement of the deeper properties of tissue. However, that also means we are measuring from a larger volume without spatial resolution. Another technology that's commonly used are spectral imaging technology. Usually this means we expose the brain and use a camera to take images under different discrete wavelength illumination. This would provide us with really fine spatial resolution, but this measurement is usually only limited to the brain surface. So from this two, we can conclude that from the intrinsic signals, we either measure from a small volume on the surface with fine spatial resolution, or we are limited to the surface. Basically, we cannot do deep brain and high spatial resolution at the same time. So that's the research questions that I was targeting. How to measure from a small volume of tissue in the deep brain. Another focus that we want to address is the measurement from freely moving animal. This is important because this allows neuroscientists to study the animals in a more natural state. And studies have shown that anesthesia can alter the normal physiology of the brain, and restraining the animal induces stress, which can also change the physiology and the behavior. So there are two types of systems that can enable this kind of measurements from freely moving animals. The first kind only miniaturize like the head-mounted part. For example, this is implanting a fiber, or this is like implanting a miniaturized microscope. However, like it still has like connections. It could be fiber or wire connections to a benchtop system for data acquisition or for power. The second kind of system like miniaturize everything, so the animal is tether-free and can run around with it. So this type of system requires more engineering work, and it's much harder to design and test. So the objective of this study was to develop a novel optical tool to study the brain using intrinsic optical signals. We are targeting hemodynamic measurements from a highly localized volume in the deep brain, and also from free moving animals. And our solution is single fiber spectroscopy, which would provide a wavelength resolved measurement from the line of sight region in the brain. So the idea we have here is really similar to fiber photometry or optogenetics. Both of those are really commonly used in neuroscience studies already. The idea is to implant a small diameter fiber and target it to the region of brain, targeted to the brain region of interest. So here, like for example, the fiber would be implanted to the tissue. It would illuminate part of the tissue. Light will be delivered here. After some interactions, some of the light which has been scattered and absorbed will be collected by the same fiber. In this study, we use a 200 micrometer diameter 0.39 Na fiber. This is chosen based on the sampling volume, because we are targeting small brain structures, and the sampling volume, depending from the Monte Carlo simulation, shows that we are sampling from a 0.02 to 0.03 cubic millimeter region. Here it shows like schematic of the system. It's a pretty straightforward design. We use an LED as the light source. The light is collimated, and 50% of light would pass through a beam splitter, being focused into a multimode fiber and get into the tissue. After the interaction from the tissue, the signal light will travel back, and 50% of them will be reflected towards a spectrometer for detection. One thing that we want to note here is that the reflection from the fiber surfaces will also come back the same path and be collected by the spectrometer. And this would decrease our dynamic range, because the signals are not, this light are not the signals that we want, which has interacted with the sample. Such that we engineered the system a little bit differently. What we did was to angle polish the fiber surface, such that the reflected light and the signal light that comes back will go towards different direction. And in this case, we avoid the reflection from going into the spectrometer and have a better dynamic range. The fiber far end here are also polished at an angle to reduce the reflection at the fiber brain interface. Simulation shows that when it is 24 degrees here, the light from the fiber reflection will not go into the spectrometer. So just now we presented the system, and it can measure the signal light from the tissue. So how to extract the biological parameters that we want from that signal. Here we use an empirical model of single fiber system for this. So this is a model which relies on the empirical Beer-Lambert law. The basic idea is we have a fiber here which is in contact with the tissue. When there is no absorption in this tissue, light gets scattered back, and some of the light can be collected by the same fiber. We assume that when there's no absorption, the signal that can be collected are R0. Then when we consider that this tissue is absorptive, some of the scattered light will be absorbed in this process and won't be able to be measured anymore. So if the signal here are RSF, when there's absorption, we can have the equation like this. This exponential term is basically based on the Beard-Lambda law that consider the attenuation due to absorption. So here, mu A is the absorption coefficient of tissue, and LSF is the path length. Basically, what's the average path length that the photons has traveled inside this tissue. And this LSF parameter depends on both the absorption coefficient and scattering coefficient. And we basically adapted an empirical equation on that. And with this equation, we can fit into the spectrum and find out what's the absorption coefficient, and use the absorption coefficient to extract the biological parameters we want. So first we evaluate the system performance in an optical phantom. The basic idea is we use a liquid phantom, which mimic the optical properties of the brain. And we use both the single fiber system and also a near-infrared spectroscopic system, a NIR system, as a reference system to compare the results. We controlled what's oxygen saturation inside this phantom by controlling the gas content inside this box. And this is depending on the oxygen dissociation curve of hemoglobin. Here are some spectrum from this phantom experiment. On the left side, we see like when the blood volume fraction is 2%. And at different oxygen saturation, we can see like the shape of the spectrum changed differently, and or measured and fitted spectrum. The fitted spectrum was quite well, which captured most of the features in the spectrum. For 2%, for 3% blood volume fraction, if we compare, we can see like the amplitude of the signal are lower than those in the 2% signals. And that's because when there is more blood, it absorbs more light. So the basic concept, again, we are using the absorption features here. The shape of that tells us what's the oxygen saturation. In other words, what's the ratio of hemoglobin that carries oxygen. And the amplitude here tells us about how much blood exists in that volume. So with this two, we can figure out the oxygen saturation and blood volume fraction. Here is a comparison of the results from the two systems. The single fiber system compared to the NIR system. So here we are not, because there is a lack of gold standard, we have to use the NIR system as a reference. But again, it is also an optical system which hasn't been characterized with a gold standard, therefore, we are only going to make conclusions that SFS, the single fiber spectroscopy system, predicts the correct direction of oxygen saturation, and we're not going to read the accuracy. After the system is done and the initial calibration process in the phantom, we optimize the system for fitting-moving experiments. Here, again, we adapt the concept of optogenetics and fiber photometry. So we have implantation Faru here, which will stay with the animal. This implantation Faru is implanted to the region of interest a few days before, such that the animal can be returned to its home cage and rest and recover before the real experiment started. When we do the real experiments, we will use a patch cable, another fiber, to connect the animal with the benchtop system. The alignment is ensured by a mating sleeve. And again, all the fiber interfaces here are polished to reduce the back reflection. Here are some photos of the Farus that we made. So again, like here shows the polishing, angle polish surfaces. And here is a photo showing the animal with the implantation Faru, and when it is connected to the patch cable. So firstly, we started our longitudinal studies. So over 31 days, we measured the oxygen saturation in the animal. So the fiber was implanted on day 0. In this case, we put the animal down with under anesthesia with isoflurane mixed with pure oxygen. And here it shows the trend in oxygen saturation when we switch the animal from breathing pure air to room air. So as expected, we see a change, a drop in oxygen saturation when we switch this gas content. And the shape of this change are very similar in the same animal over the course of 31 days. And this shows us that SFS can measure the oxygen saturation values robustly in a longitudinal study. For the next study, we started doing our freely moving animal experiments. In this case, we put the animal in an airtight box and change the oxygen saturation, change the oxygen level inside this box. And we are hoping to induce a global change of oxygen saturation in the brain. And here are two videos showing the animal walking around with the fiber connected. And the system is now like, at the same time, the system was measuring spectrum while the animal was freely moving. So we did this experiment without using a rotary joint, and that's why this fiber got tangled after 1 hour, but it doesn't affect the signal. And we were still able to extract the hemodynamic parameters from it. This figure here shows the oxygen saturation level measured by the single fiber system, which is in the black curve, and also what's the oxygen level in the box at the moment, which is this red curve. So one thing to note here is that the black curve is oxygen saturation, which is how much oxygen is carried in the blood, and the partial pressure of oxygen is basically a description of how much oxygen is in that box. These two are not directly related because the animal breathes in the oxygen, and also there are brain activities that's going to induce change in oxygen saturation locally in the brain. However, here, if we look at the trend of the signal, since we're controlling the oxygen saturation, the oxygen level in the box, these two align pretty well if we look at the general features. One slight difference here is because we were putting the animal down under anesthesia, again, mixed with pure oxygen, such that like the animal was breathing a really high percentage of oxygen and having a really high starting point. But other than that, the trend of the oxygen saturation and PO2 match pretty well. And this was the first demonstration of hemodynamic monitoring in the brain from a freely moving animal. So yeah, so from this presentation, I presented the system design of single fiber spectroscopy. There were some Monte Carlo simulations to find out what's the volume of the that we are targeting using this system. And also we did phantom experiments to find out what's the accuracy or like how reliable the results was. And we optimized it for freely moving animals and demonstrated in some in vivo experiments. I would be happy to log into, like to dive into any of the detail later on. So that's all for the single fiber spectroscopy. And I would like to help to thank everyone who have contributed to this research and also my funding agencies. Yeah, thank you.
The student volunteer part. If you
sure,
if you have the energy.
Yeah, I mean, like, it's just like I need to have a hard stop at 3 because I have a meeting afterward. So I wanted, okay, like, maybe we just want to open the discussion directly. So basically, like, I guess I'll describe a little bit about like the OSA or the Optica ambassador role. What is it? And how I may be able to, you know, help or support you in any way. So basically, like, the Optica Ambassador is a program that Optica, or the formerly Optical Society, that we have. It's like we have a group of early career professionals every year to talk to students around the world in different student chapters and, you know, in different regions. And we are here to share or advise or experience and see if there's any way that we can support you through your like career decisions and, you know, all different considerations in like your career. So basically, like that's the other role that I have. So the other presentation I prepared is really simple. It's basically talking about my career path, but due to time, maybe, you know, like we're just going to, I think I'll just open the floor here. If there's anything that I could help, or if you would like to learn more about my experience, I can share that as well. or if you have any questions regarding like, grad school, postdoc applications, job hunts, how to get involved with OSA, I'm here to chat, or like you can reach out to me anytime. Yeah.
So I could
good opportunity searching for postdoc. I'm almost wrapping up my PhD. I think